Okay, good morning, everybody. And maybe good afternoon, depending on your time. So I hope you grabbed your coffee. So I did, actually it's not a coffee, it's a tea. I had my enough coffee last week. I've been in Italy. We are going to have a break in your work to ask questions to me about TRANSFAC. So welcome to this session. So we'll make these sessions more often, I hope, in the future, because so far we got quite a lot of questions, and I have prepared some answers to some questions we are getting repeatedly. But of course, you're welcome to give your questions to ask me right away. So please put your questions in the Q&A section. So I will see the questions and I will answer them, try to answer them. So this session will be maybe for 1 hour. Let's see how it will go. So as I said this, let me share my screen first, what I have. Okay, good. So actually, there are, as I said, there are a number of questions, and I have collected them and maybe grouped them a little bit to start from the very beginning. But let me first introduce a little bit what we are talking about. What is TRANSFAC? Although if you came, probably you already know, but just to refresh a little bit of your memory, what we are talking about. So just a few slides. Basically, TRANSFAC is developing by GeneXplain company, which is located in Wolfenbüttel. It's a small town in North Germany. It looks like that. So you see, very nice town, and it was mainly founded by me and Professor Edgar Wingander, who is a major developer, a major sort of initiator and creator of TRANSFAC database. And so we continue to develop it, and actually from the beginning, it's in fact 30 years. So you see on my cup, it's 30 years of TRANSFAC. So actually it's more now. It was 30 years, I think 3 years ago. So it's now like a very mature database, 33 years. And so our company is not only developing TRANSFAC, which is mainly concerning the bioinformatics analysis of regulatory regions of genome, but we are dealing with other topics, and we have databases and software in all these topics which you see here in systems biology, also in systems medicine, and in chemoinformatics. And I hope in my next sessions we can touch those topics as well. Although you are free to ask me questions about any of those software which GeneXplain is creating and distributing. But we start from TRANSFAC today. And TRANSFAC, it grows from, as I said, from several years. Now we are talking about TRANSFAC 2.0, which is kind of a new reincarnation of a TRANSFAC, which gets a lot of new software attached to the TRANSFAC. But basic is the same. So basically, our team is going through literature and through high throughput data, and create a database which contains information about transcription factors. So the proteins which are functioning in the nucleus of the cells, mainly concerning actually transcription factors of eukaryotes, and mainly focusing on such organisms like a human, mouse, and rat. But not only. So we have information about transcription factors from other eukaryotic organisms, and even from plants, but not really extensive, I would say. Although in the field of plants, I would say TRANSFAC is quite unique resource. About the transcription factors, so that's kind of a lot of proteins in human genome, I would say, and our estimation is it's around maybe almost 2,000 genes in genome, which I encode various transcription factors, and we classify them in classification of human transcription factors, which helps to understand sort of classes of factors and how they function in the genome. Let me check. There is one chat question here. No, that's just telling you again that please put your questions in Q&A session, Q&A sort of button, and we'll see these questions and I will try to answer them. Okay, so but major, I would say one of the most questioned and requested assays in TRANSFAC are these positional weight matrices. And let me explain a bit what it is. So what we are doing, we're going through the literature, as I said, and collecting information about individual binding sites for transcription factors. So for, let's say there is AP1 transcription factor, and we go through the literature and find binding sites which are revealed by experiments, various types of experiments in various species, human, mouse, and rat. And then we align those sequences, those binding sites, and we build this positional weight matrix, which is a representation, some sort of model of representing the profile, the binding profile of these factors to the sequences. And you see it's very simple. Basically, we count how many times a particular nucleotide is found in that particular position of that site, and then we normalize it to the number of sites which were considered, and you see if this value is very high, it means this letter T in the first position is very frequent, but there are some other letter may happen. So you see a lot of variability. So, and there are positions which are really quite variable, like this one. You see it's well, although some nucleotides cannot be found there, like T in that position, but others may found be also. So this positional weight matrix is, as I said, is a model which is used then to predict new sites. Why do we need to predict new sites? Because not everything is known, and we are very far from knowing everything, from knowing all binding sites in genome. And even with ChIP-seq technologies and actually floating us with all these binding profiles from various transcription factors, which were detected by these ChIP-seq and more exact variants of these technologies, still give us huge amount of binding sites. But according to our estimates, if you assume how many sites are actually functioning in genome or can function in genome, that could go up to 2 orders of magnitude higher than we still know, even taking into account ChIP-seq information. So that's why we still need to predict binding sites in genome, and that's how this method works. And that was actually one of the questions repeatedly asked by our users, how this method works. And let me explain a bit again. So what we do, we take this matrix and take a sequence of our interest, and this matrix is then scanned through the sequence, and in each position of the sequence, we calculate this score. This score, and the score is in fact very simple. It's you have a particular sequence in that exact position, and then in that when we calculate the score, we multiply or sum up, I would say, the weights from that matrix, which you get depending which nucleotide you see in that position. And that weight is multiplied by a factor, which is called information vector. And that's very interesting, actually, contribution, which tells that if this nucleotide is very conserved, which means is very frequently found in that position, it has a high information content. And then this information is multiplied by this weight, which means in the important positions of the site, we kind of giving additional weight to those frequencies which were observed. And that's some special method, which is, I would say, sort of unique in the MACH software, which is associated with TRANSFAC. So there are other types of methods which can calculate this score, which are used by other software, but using this information content is somewhat special for MACH software, and maybe a few others, but not really widely accepted. In our experience, this information content really gives emphasis to the conserved nucleotides in the sites, and it works in many cases better than just summing up the weights or in some logarithmic scale or some other methods. Actually the preferred methods is depending on your wish, and we recommend this method, which is using this information content value. So once this score in the binding site position is higher a given threshold, then we call this site. We are saying that in this position we observed the score which is higher a given threshold, and this might be a binding site for the transcription factor of AP1. So we predict that this site can happen, can work, but in which conditions we didn't know. It actually gives us just potential that this place could be a binding place for AP1. And actually, to go a bit more in details, there was a question concerning, but maybe we'll come to that one later, because we need to explain a bit how these scores, how the cutoffs are calculated. But let's first maybe look at our list of questions, and also if you have your questions, don't hesitate, just put your questions during my talk, and don't wait until the end. So, we will see the questions. So let me see. So the first question was, how can I start a TRANSFAC bioinformatics analysis, and what types of data do I need? So I think what the question means that the TRANSFAC bioinformatics analysis is, I guess, what the author of that question meant, that the analysis to find binding sites for transcription factors. So exactly what I explained. And this is done by a special software which is attached to TRANSFAC. It's called MACH. And that MACH software is incorporated in different variants in our tools. So let's have a look at our website where you can find this MACH software. So let's go to geneXplain.com. You see, there is a TRANSFAC. You can read about TRANSFAC, but to start your analysis, you have to log in, and for that you have to have a license to use TRANSFAC. We have various models of licenses. If there will be a question or there will be some time, I can explain this a bit later. But once you have your license, you have your account, you have your login credentials. So you can log in one of our software benches. Sort of the first one which we want to consider is this GeneXplain portal. So it's a portal to look for information from the various databases which we provide, also from TRANSFAC. And we will start from that. So let's log in that portal. So we have this, I have this login prepared for this example. And as you can see, this particular login opens just one database for you. So as I said, depending on your license, you can have access to all of those databases and software, or just to one of it. So in this case, this user has access to TRANSFAC. And in TRANSFAC, you can have, yeah, let's have a look in TRANSFAC interface, which allows you basically to search for information about binding sites and about transcription factors and so forth. So let's, for instance, search for NF-kappa B transcription factor. So you see it gives you information that in our database, in TRANSFAC database, there are 32 matrices which concern with NF-kappa B transcription factor family. And there is 71 transcription factors for various species which matching this name. So let's look at those transcription factors. You see, for instance, NF-kappa B1, that's a gene name. It's a protein name of that transcription factor, which is from Homo sapiens, from human. You see the name, description of that factor, and what we can do, we can look at that entry to see more information about this factor. So there are a number of questions coming now. So I see the questions. Okay, I will come to that questions in a moment. Let me finish with that. Let me finish with the NF-kappa B entry. So what is important in this entry? So you have, of course, information about the protein itself, what is kind of function it is belong to. It has the information about the whole orthology hierarchy. You know, if you just rely on the name, it may be misleading, because in different species, this protein or this ortholog to that protein may have different names. But in this case, this is not the case. Well, it could be variations of the names. So you can navigate through these names depending on the species you're interested. Then there is actually an information about this transcription factor leading you to another database, human gene mutation database, about mutations in that protein which are relating to different diseases. And this is a very important resource. You can click, and if you have a license to that database, you get information. So here we have just statistics saying how many mutations are observed in this protein, which were related to various diseases. Then what is important, and we will talk about that a bit later, that actually this factor, as any protein, is expressed differently in different tissues of human body. And you see the relative expression of that protein in different tissues. And for instance, you see in lymph nodes, it have a maximum expression. In bone marrow, it has a maximum expression. That makes sense because, as you may know, NF-kappa B is a transcription factor which is involved in various function of immune system and inflammation. Well, and you see those cell types and tissues which are related to the immune system. And you can also get a bit more details about which tissues you observe the expression of that factor, and that's collected from various information. This information is collected mainly from Protein Atlas, and this type of information about individual observation of expression of that protein in various tissues is collected from various literature during through our curation process, as you can see here. So another section for that protein is, in fact, this protein is encoded by some gene, right, by an NF-κB1 gene. And this gene, in turn, is regulated by other transcription factors. And for instance, this gene is regulated by NF-κB by itself. So it has a certain feedback loop. So it expresses this protein, which is able to regulate expression of its own gene. You see this binding site, and this indicates the binding site for NF-κB transcription factor. But not only so, you see there are other sites which are known for that promoter of that gene. So there is a site for other factors, and again another binding site for NF-κB transcription factor. Here we see another site, so it's really heavily self-regulated. You have here below you have also information about all other binding sites in the. So actually, it's just the same binding sites, but more in tabular form. So you see here 16 binding sites, known binding sites in the promoter of NF-κB gene. And we have a separate table of information about ChIP-seq fragments for various transcription factors which are observed in the regulatory regions of that gene. And you see there are quite a number of ChIP-seq experiments were done where this gene was target of various transcription factors, and you see these names of factors and where they are binding. So according to the ChIP-seq data. So again, what is the difference? And that's important to understand. So these 16 binding sites are, I would say, the classical binding sites, which means they were identified by classical experiments of gel retardation, of mutation, hypersensitivity of of particular regions of promoters. So the classical methods, which can be, we have actually inside also a classification of those methods telling this particular site how well it is identified by maybe by one method or by various methods. But for instance, this particular binding site in that promoter was identified by various methods. Not only it's done by functional analysis, but gel shift assay, by super shift, and the quality of evidence that this is really true, experimentally known and functional binding site is quality 3. And we have actually 5 qualities. Quality 1 is the best quality, which means the site is confirmed by various experiments, and it's also known that this binding site is actually functionally important. It's really driving this transcription factor, transcription regulation of that gene. And quality 5 is the worst one. And in case of and these are classical binding sites. But as I said, we have a second table for ChIP-seq revealed binding sites, which are also, of course, experimentally verified. There are some sort of drawbacks, or not saying drawbacks, but not completeness of evidence in case of ChIP-seq, because first it just reflects the binding affinity of that factor to that place, but saying nothing about whether it's functional, whether it's really involved in regulation, this particular binding site. And also the regions of binding sometimes is not well defined. So the peak of the ChIP-seq experiments can be very wide. With some special methods, you can make it more narrow and basically come to a really narrow place. But quite often you don't know where exactly this binding is occurred. So, but still, it's very valuable information, these ChIP-seq binding sites, and you can see it here in the entry. So there is more other information quite a lot of information about which other factors this protein, this NF-kappa B factor can interact, which which pathways it's involved, and many other things. So let's stop here and maybe look at our question. Okay, so the first question what I see is, what is the significance of a transcription factor binding sites on plus or minus strand of the gene? Thank you for that question. And that's actually a tricky question, and sometimes really misleading for many people. But let me simply look at one of the picture. Let me see if I have such a picture here. Yeah, for instance, this one. So the binding sites in genome. So actually, the transcription factor, which is like can be one protein or a complex of proteins, like for instance, AP1 is in fact a complex of two proteins from C-Jun and C-phosphamides. Very tight complex, and they act as one transcription factor. But most often this is just one protein, and that protein has a DNA binding domain, so which means the place in that protein in 3D structure, which in fact touches the DNA and binds to it. And DNA is, as you know, is double helix. So the protein, it's in fact binding to the DNA, which is have no symmetry. It has a symmetry, but in 3D world, it's like a tube, and the protein is binding from one side of that tube, I would say. Right, and some of the transcription factors they bind into the major groove, so this big cave of that DNA structure, and some of it to the minor groove, and some actually binding very complicated ways, so in touching this DNA in various positions. And here beneath you see there is actually a sequence of that binding site, and it has a certain symmetry, right? So for instance here the plus strand is this is plus strand, we call it, this is the minus strand. And the protein can bind in a certain one direction, so we can say it's coming from the plus strand, but it can bind from beneath, from the bottom strand. And in fact, in the whole geometry of the cell, it does matter. It's just a tube, right? It gets matter. It's become important where exactly is the start of transcription, or where exactly is the gene, which is then would be regulated by this transcription factor. Is it at the right place or at the left place, right? And then once you know where the start of transcription, where the gene is, then it's getting important sometimes the directionality. So whether the binding is occurring from top or from bottom. But if you don't know which direction regulation should go, in fact, you cannot identify what is the right direction. And the question was actually whether it's significant, whether it's plus or minus. So plus or minus depending on the start of transcription, as I understand, right? If you don't have a start of transcription, there is no plus and minus. So if you have a start of transcription and the gene is actually encoded from one strand, so it means this is a plus strand for that gene. And the opposite strand is a minus strand for that gene, but it could be a plus strand for another gene, which is at the left side, right? So, and then if you consider the gene, and for some factors, it's indeed important, the directionality. For instance, we know the TBP, it binds to Tata box, and Tata is just, when we say Tata, it's sort of symmetric, but if you consider the more contents of that Tata, it's not symmetric, not fully symmetric, which means this transcription factor should be, this binding site should be on plus strand. But there is another another sort of puzzle here, or this kind of puzzle. So the point is, on top of this real, functional category, there's also artificially created problem. The problem is that when we build a positional weight matrix, we actually have to choose the orientation of the binding site, right? And it just by random chance, we choose one or another orientation. And then one certain matrix can be built from all sides which are collected, I would say, from the plus strand. And it could happen that for the same factor, which in fact binding from the top, we collect binding sites from the minus strand to build the positional weight matrix. So which means there'll be two matrices for the same factor, but they will look completely different, because one matrix was built from the plus strand and another from a minus strand. But in reality, it's the same factor, right? So it's the same thing, which means when you apply the matrix in the match algorithm, then you will see this error on the prediction, like I showed just in a moment. But this error doesn't mean nothing. Actually, it just means this is orientation of that positional weight matrix according to the orientation how it was chosen when we built the matrix, or when because we are not only building those matrices by ourselves, but we are collecting matrices from other resources, from the publications, from databases, when people analyze, for example, ChIP-seq data and build those matrices, and they build those matrices often using automatic software, and this automatic software choosing randomly the orientation of the site, and it may happen that one matrix for NF kappa B is built from plus and another from minus. So the bottom line, I would say, it's not really. You shouldn't pay too much attention to the arrow direction in that prediction of a binding sites, because of all those things which I said. Rarely it may have a sense, but I will maybe talk about this later when we talk about compositions of binding sites. Because when we talk about compositions, we are now again getting the directionality, because one side could be on the left side and another on the right side. And then it could happen that the direction of one side in relation to another side is important. And in some cases it's important, in some cases not. Okay. So another question, the question about transcription factors. Do you surely survey that all the time? How many targets are necessary for inclusion of creation of a matrix in the program? Okay, right. So what you mean is that basically how we continuously building our positional weight matrix library, and indeed we have our curators and certain number of curators, so we are not gods. We cannot collect everything which is published, but we are doing our best. So continuously looking in the PubMed and all the resources which are available, and continuously taking all the information about proven binding sites for transcription factors. And also in the current world, in the recent world, we get a lot of information from ChIP-seq experiments, and so we also not ignoring them. We collect, as I showed, ChIP-seq data in our database, and also using it to build position weight matrices. So in case of ChIP-seq data, your second part of your question is not relevant. So basically, how many sites do you have to build the matrix? In case of ChIP-seq data, usually there are tens of thousands of binding places in genome, which are observed in that experiment. And then there's actually rather an opposite problem to reduce number of targets, maybe to the most significant ones, to build your matrix, because as you know, another problem of ChIP-seq is that it has a certain experimental error rate, so there might be quite a big number of false positives, which means factor for whatever reason or the experimental reason was found to be bound to that place, but it actually is not binding to DNA. It may bind to another protein which was sitting in that place, and that's why you will not see the motif in that region. But anyway, in case of ChIP-seq data, we don't have this problem of collecting enough sites. When we built the regular positional weight matrices, and that was mainly what we did in the previous 30 years, was to collect enough. And our rule was that it must be at least 10 binding sites for from various targets, or even from the same gene, but maybe several places in that promoter, so that also would count, but must be at least 10 sites. And then we built a positional weight matrix from that number of sites. If it's less, sometimes we do, but very rarely. So only in case if those bindings are really binding sites are very, very similar. There's a very little variability. And the reason why we need a certain number of sites to be sure that we build a correct matrix, that actually, I touched it a little bit, but that's a big issue and also a big property of transcription factors, which often is ignored, but it's an important property, that transcription factors and binding sites for them are very heterogeneous. So, which means you have really to collect enough binding sites to capture maximally that heterogeneity of those sites to build a good model to predict the sites. And why they are so heterogeneous, that's a topic for another big discussion, which we can have. It's more towards evolutional considerations and so forth. Okay, so that's my answer. So another question. A PhD student working on transcriptomics in astrocytes biology with in Nensk Institute. Actually, I know your boss. Actually, I met her last week. Okay, I have creation quality of TRANSFAC database, and I wanted to ask a few questions regarding TRANSFAC. What advantages does it have over the HOCOMOCO and JASPAR databases? Okay, that I will just come to in a moment. Also regarding transcription factor analysis, transcription factor binding analysis, even after we get putative motifs for TFs, how many TFs could possibly bind at a single site on the genome? Oh, very good question. Depending on the significance cutoff, I find the TF binding sites from 2 to 3 to almost 4 to 50 at the same location. How do we filter this noise or get accuracy result? This question puzzled me from the very beginning, and I'm thinking about this already 30 years or even more, and I have some answers to that. And we have actually tools already ready to answer this question. But let's first touch this TRANSFAC versus HOCOMOCO and JASPAR databases, and that's quite often asked, and we have prepared a comparison topics to that topic. I'm wondering if I have it in my hand. Let me see. Okay. So I have opened it recently. Yes. Actually, we recorded several videos, and one of the video was just on showing advantage of a TRANSFAC database over JASPAR on one example. Of course, you can say you can always find such examples, but just to that's important to emphasize one important thing. TRANSFAC is often criticized, sort of, that we have a lot of positional weight matrices. Like, in fact, I didn't show the statistics in the beginning. We have 10,000, even more now, positional weight matrices collected in TRANSFAC. For vertebrates, it's more than 7,000, like around 8,000 positional weight matrices. And as I said in the beginning, if you consider human genome, there might be around 2,000 genes, or maybe less, a little bit less, encoding for transcription factors. But we have 8,000 matrices, which actually clearly say that for one factor in our database, we may have 2 matrices, 3 matrices, almost in some cases 20 different positional weight matrices for the same factor. And that's often a source of criticism, because people saying, okay, why you have so much of redundancy? And some databases, and initially JASPAR was focused on selecting one matrix for a factor, the best one. Now they also extended, so they have various matrices for the same factor. That's obvious. Why? Because in fact, as I showed in this video, that well, if you start interpreting your data, for instance, in this case, we were analyzing SNPs from COVID-19. So there is a number of SNPs associated with severity of that disease. And when we analyze one SNP, which has a biggest impact in that severity, you can see the tools which we provide can check which transcription factor binding sites are actually affected by that SNP. And what we found that actually this binding, this one SNP is create a new IRF3 factor binding site. And that opens the whole field. Basically, we may say now what kind of pathway is involved in that severity of that disease, because we found that binding site, and we can further speculate how this binding factor, because IRF3 is involved in regulation of immune response, and why this SNP can be so important. Well, when we applied JASPAR, we couldn't find that binding sites. Not because JASPAR is bad, but because simply this particular matrix was not there, was not present in the collection. And that emphasizes the bigger your collection of matrices, the more chances you have to find facts which are important for the regulation of your genes. And since we started much earlier than all the databases you mentioned, we have more positional weight matrices collected in the meantime. There's another angle here, which is a little bit of funny. Basically, as you know, in the public science, there is a lot of competition. So one scientist created a database, and he wants to have it, better and better than the competitive database. To a matter of fact, in the commercial world, like we are, there is no competition. So for us, we have no honor if this is our matrix or matrix from another database. We simply collect everything which is available, which means we collect matrices from JASPAR and from HOCOMOCO as well. So they all are in TRANSFAC, plus what we collect in BioInformatics. Or if you have other databases available, we also collect from. And we compare those matrices to each other. We remove redundancy in the sense that when the matrix is exactly the same, We remove really bad matrices. We do proof kind of error pruning of matrices. So for us, important is really to provide a tool which is as best as possible. And whether it's our matrix or matrix from another efforts, it doesn't matter for us. Yes, that was concerning the comparison. Let me see. Yes, this is very important question concerning the how many binding sites can be found on the same place, and that's yeah, there is no quick answer to that, but let me at least touch that topic. Let me open what I prepared here, because I was expecting your question, actually. This is very frequently asked question. Just to, I have such a picture where what you mentioned, just to, to stress you a little bit. And to you are talking about 40-50 sites on the same place. Let me see if I have here similar pictures. Probably not, but okay. Yes, indeed, you can have many binding sites on the same place. And earlier, people thought it's a kind of artifact of your predictions. Right? How can it be? But now people know, and that's the nature. And what you see in fact on the screen is experimental results. So it's not predictions; it's experimentally, it's experimentally proven binding sites for different transcription factors. Let me explain. So that's one region in genome which is an enhancer of a MYC gene. And that's a very important region. If there is one mutation in that enhancer, it leads to more frequent colon cancers. But anyway, so that's enhancer. It's called even super enhancer in some places. It has several megabase lengths, and all these dots, and this is a little bit zooming of that smaller region. All these little arrows, black, are the ChIP-seq peaks of many different transcription factors. And this was done in the same cell line. So it's not coming from different cell lines or from different situations. It's coming from the same cell line. And as you can see, if you take one place, so several different transcription factors can bind to that place. And they are actually really bound. So of course, this is not a single cell experiment. Maybe in one single cell, only one factor is sitting on that place. That's a bulk ChIP-seq, so which means it's coming from millions of different cells, although they are coming from the same cell line. But obviously, in that even cell line, in different cells, this region is occupied by different factors. Why? That's a big question why it's in one cell line. But if you talk about different cell types, different tissues, then it's becoming clear why the nature makes such a thing. Because in liver, this gene has to be regulated by NF-kappa B, or by, for instance, HNF4. In immune cell, the same gene, we have the same genes in all cells, but in different cells, they have to be regulated differently. So the same place should be occupied by another factor, because in that cell time, this particular factor is playing a role in regulating that. And why? Because, we have too few genes. We have in our genome just 20,000 genes. And how many situations can you expect that genes has to be regulated? There are millions. So in various cell types, in various conditions, you press on the cell, you put some drug on the cell, you hit the cell, you put the cell in Mars, or in any condition which was even not expected so far. In all these conditions, these genes has to be regulated differently. And that's why the whole promoter of that gene, the whole regulatory regions, it's a kind of puzzle which can be occupied by different transcription factor binding sites, transcription factors, which means your question, which is making not real sense, in the sense how we can filter the noise. There is no noise. These all are, well, not all, maybe there is some noise, of course, but most of those things which we predict, they are functional. So they can function. This place can be targeted for binding of a particular transcription factor in case if those conditions will come. Which means they all are functional. But now, still, you are disappointed. What can I do with all these millions of sites or thousands of sites on my gene? And then important is really to understand what do you want to do next. And the next, often you want to understand the regulation of that gene in particular situation, not generally in any situation, but in your particular cell type, in your particular conditions. Only in that case, you are interested which binding sites are active in that promoter in your particular conditions, right? And we have a tool which can help you to do that. This is based on genetic algorithm, and this tool is finding combinations of binding sites. So out of all these 50, as you said, the same place has 50 predictions of different binding sites. But now if you put in the input of that software a set of promoters of genes which are regulated, for instance, upregulated in your conditions, then this algorithm will work for a couple of hours and it will find the combination of binding sites. It will pick up from those 50, one or maybe two, which are important in combination with your with other factors, and they together regulate your gene. So let me show a result of that tool. For instance, in this tool can be applied not only for promoters of upregulated genes, which is a major use of that tool, but it can be applied also to any functional regions. For instance, here we apply it to analyze regions coming from ChIP-seq experiments in particular cell types. So we want to know where a transcription factor called E2F is binding. So this we took this data from ChIP-seq data, and we put those ChIP-seq regions in the genome, asking a question: if E2F is binding to that place, what other factors are cofactors for this E2F, which make this place special? So you know you have a whole genome, but for some reason in that particular condition, E2F is binding to these 1,000 places. So which means E2F binding site, E2F binding motif is accompanied by other motifs or other factors, and they together in a complex, they make this binding specific. So one factor cannot make really specific binding. In rare case, it can. For instance, there are very important and famous factors like this TCF. Wait a moment. So the insulator factor, I forgot the abbreviation. So this is a huge factor. It's a huge binding site, and it's really making by itself very specific binding. But the regular factors which are involved in gene regulation, they are actually, as I said, binding sites are very small and very unspecific, and that's why one factor cannot make specific binding. So it has to be always a complex. And this program, what it shows you, you see each line here, and it goes down 1,000 lines, but here one line is one place of ChIP-seq region, and the program found it's one ChIP-seq peak, and the program found these transcription factors cooperate together, and you see the binding arrows for each of those factors, and you see E2F has to go together with ETF, ATS, SP1, AP2, and this graph shows you that if you compare any random region of genome, actually which contain, in this case we also took regions which may have E2F binding site, single motif inside, but single motif doesn't work. So this combination of motifs discriminate all these ChIP-seq fragments from all the rest of genome and make it really specific. So that's the answer to question how to filter. You don't need to filter, you have to find combination. All right, okay. There is one question in chat. There is also a link to the YouTube, so you can watch this, what I'm saying now in the YouTube. Good, so somehow I answered. I didn't answer yet. The last one. So this I answered. Okay, good. So now, is it possible to use directly gene sequences to search TFBS? For example, sequence from Ensembl database. All right. Yes, of course. Sorry, I was preparing this from the very beginning to show you what kind of data you can use. And let me show this. This is really important, but I start talking and I missed that important point. So what kind of data you can use to start your analysis? And let me show you that just in examples. Let me simply open our tool, which I didn't open yet. Yes, right. So Ensembl database and FASTA is both possible. And let me show you how we do that. So to analyze your data, as what I showed before was the database itself. But now how to analyze your data. So for that, the number of tools, and there is one tool which is called Match, which I touched a little but didn't show yet. But there is the whole platform, we call it GeneXplain platform, which contains all of those tools. It's really a platform to do your studies. So as you can see, this platform contains tools and workflows. It actually has more than 100 different workflows and even more different individual tools to analyze data in various formats. And for instance, you were asking for Ensembl format. So basically, you go here and you load your data, and you can load it in the form of table, in either Excel table or simple flat file, which contains in the first column, it contains basically accession numbers from any of the databases which you can name. So some of them are mentioned here: Ensembl in the first place, GeneSymb, UniProt ID, IPI, RefSec, and many other IDs it can accept. So just to show you an example of that file, let me. I have prepared this in my examples. So for instance, this is an example of input file. It's actually, this is an Excel file, and you see in the first column, there's Ensembl IDs, and it can be many other columns, and the most important to have this ID column in the first column, and then you can have fault changes, you can have p-values, you can have various different values which can be used in the further analysis. And by the way, you can also use various accession numbers from various resources. For example, from microarrays, from Affymetrix, Agilent, and we can also accept those accession numbers. It will automatically convert them into the genes. So, and once you have such a table, then you simply load it into the into your folder. But let's first open your folder. It's open here in the tree. So you have your data folder where you can create, for instance, a new subfolder where you want to start analysis. So let's call it example 1, and then you click on that folder, and then you load your data, either clicking here, or you can simply click on import interface, and you can import your data from various sources. You can basically take it from computer. I will show in a moment, but you can also take it from a repository from other place in the same platform, or you take it from FTP site. So and that's very useful when you want to analyze your data which already stored, for instance, in gene expression omnibus. You simply put here the link to that table, and it will take the data. So in our case, we want to take it from a table in our computer. So there is this Excel file, and we have to. Patients are here. You have to open it first. Loading it, recognizing it as a file. And here, by the way, you can see what type of formats it can recognize. It's not the full list. It's a pre-filtered list according to the type of the file you are importing. And it could be a table, but it could be also, for instance, FASTQ files. So raw data. It could be also generated, imported, and then there are tools to work with them. I can talk about that maybe the next time. But now it detects that this is a table, and it checks automatically, finds that the header of that table is in the first line, which could be different, by the way. So sometimes you can have somewhere else. And you define which column actually would be your accession number column. So by default, it's the first column, but it could be any column, so which you can select. And by the way, if you don't have such a column, which would be uniquely specify each line in your table, you can generate such a column by clicking here, generate unique ID. But in this case, if you have an Ensembl IDs, you have it, it automatically detects that's Ensembl, and you only click start, and then it imports and opens this table here, and as you can see, it's exactly the same table. And now you can basically analyze further data from that table. The same with FASTQ, and here you asked about FASTQ. Here we have another section, sequence analysis, and here you can, FASTA. Yeah, right. So you can load as a FASTA file, as a SRA file, or GenBank or Ensembl file containing sequence. So different formats of sequence collections or individual sequences can be loaded, and then you have to, while loading, you have to specify which genome it is, because in case of table, of course, Ensembl uniquely identifies this is a human genome. So, but in case of FASTA sequence, you don't know which genome it is, and if you want to analyze actually in context of, say, human genes, then you specify that's a human genome. But you can also analyze a FASTA file from any genome, or maybe anonymous even genome. The only thing you have to care about is when you analyze, search for binding sites in that genome, you have to select a collection of binding motifs from that particular phyla. So, for instance, if you're analyzing a vertebrate genome, you have to select collections of the matrices from that genome, from that phyla, from vertebrates. If you analyze plant genome, so it's make no sense to apply vertebrate matrices to analyze sequence from the plant genome. So you have to select the particular set of matrices. And let me quickly show you where those matrices are located. So you see we have TRANSFAC database, well, actually in various releases. If you have done something using old release, you often can be able to find it back. And then here in the profiles, you see there are various, we call them profiles. And for instance, if you want to have matrices which are specific for fungi, so you use this profile. This profile is actually a set of positional weight matrices, and their cutoffs. We didn't talk yet about cutoffs, but just to quickly say that, as you remember, our score, the search algorithm calculates the score for each position in genome, and this score can be from 0 to 1. 1 is maximum, 0 is minimum, and then the cutoff is it should be from 0 to 1 at some position at some point, and if the binding place has a score higher than cutoff, we keep it, and if not, we skip it. And there are two cutoffs: one for the core sequence and another for the whole sequence. And that's also somewhat specific for a match algorithm. It can work with this core cutoff or can skip it as well. But we, from our experience, often it's useful to have a separate core cutoff. Core cutoff is a cutoff which tells you about the 5 nucleotides in the center. And the general cutoff tells you about the whole position weight matrix. Okay, so that's about how we import data. And by the way, just to start to show you at the very in this place. So how do we analyze the data? So very quickly, concerning, for instance, set of Ensembl IDs. So we loaded it. We have this table, and we want to analyze transcription factor binding sites enriched in promoter of on those genes. So then we go here, analyze regulatory regions, identify enriched TFBS in promoters, and for instance, you can run a classical algorithm. We have, we are always update our algorithm, and in fact, if you already used classical, you still can keep using it, and if you want to experiment a bit, you can try a new one. So as you can see, I can just drag and drop here this table, and the rest is ready to analyze. And here we have scout skipping genes as a control by default, but that's not necessarily should be the case. If you have in your hands another set of genes which are not changing in this particular condition, so-called non-change genes, it would be a better control, better node set. Why do we need a control? So it will compare the frequencies of binding sites found in promoters of your list of genes compared to the random control, to see the enrichment. And then you specify the promoter length. The default is -1 kb and +100 nucleotides. And what is important is which profile you use. And here we use non-redundant just to make it really quick, which is a selection of line matrix per factor. You can search for taking all matrices, and then of course your output would be bigger, and then you have all those advantages I was telling that you will rarely miss a real site. But okay, let's run it with a non-redundant set just to make it quicker. to show you the results. So if you see, it extracted the promoters of those genes. There are 58 genes in my set, and it's around 300 genes in the no set, in the set of housekeeping genes. And now it actually shows me the results, and it opens one by one the resulting tables. Let me just wait a bit. So let me see if it's finished. Yes, it's completed. So you see here the list of all tasks which are running. And then, so the major result is here in the summary. It gives you a list of positional weight matrices which are enriched in your set of genes. And actually, as you may see here, it's called TNF stimulated genes. So if you know, the TNF is a special signal which stimulates activity of many genes, and we know that TNF is acting mainly through NF-kappa B factor, but not only. And as you can see, this real A is one name of matrices from NF-kappa B family, and it's telling you the enrichment is very significant with p-value in 10⁻13, and in yes to no ratio is 6.2, which means it's 6 times more frequent in your promoters compared to the housekeeping genes. And you can also see this visually. You can select this matrix and see in which promoter it binds in this table. So you see this gene, for example, NF-kappa B EIA, have 4 sites for this matrix. And if you click on which site, you see here the binding site, its core, and the logo of the matrix which was used here. Okay, just briefly showing you how it works. Yeah, so we are here already more than 1 hour, but let me maybe go through a few other questions. If you have time, stay with us. I still have time. Good. Okay, can I? So this one, it was already answered. Good. Either on plus strand or minus strand, one should read the binding sites from 5' or from end to 3' end. Is it correct? Yes, correct. So it's it's like that. So you have 2 strands, but one strand goes this way, another strand goes this way, right? You can also make them upside down. They are very symmetric, and in each case, your binding site has to be read from left to right, from 5' to 3'. Can a gene have 2 active promoters and corresponding TSS in the same cell? Good question. Yes, actually, what you see from Phantom, from Phantom database. So the Phantom was created by a special method called cage. When you take a cell, well, a cell, maybe cell line or a certain tissue, and you extract all the RNAs, and you mark the 5' end of that RNA with a special, chemistry, which allows you to sequence from the 5' end, and then you can map back to the genome your sequence, so you know exactly from which position the RNA was started, right? And assuming, and the cap, this important that this cage method is attaching this chemical to the cap, and cap is only possible when there's a real start of transcription, when the RNA is full, because in the cell there might be truncated RNAs, but you need the one which have a cap. So which means you know exactly where the start of transcription. And if you look at Phantom database, which was done from the same cell line, you can see that the same gene can have several starts of transcription. although we don't know, it's not a single cell analysis. It's still the bulk sequencing. So maybe among the, although it's a cell line, maybe in one cell there is one start, in another cell there is another start, and that's most probably the case. Actually, as you can see in ChIP-seq experiment results which I showed, it was also the same cell line which have multiple various transcription factors may be bound to the same place, which means they either bound to the same place in a time manner, so or in a cell manner. So in one cell, one factor, another cell, another factor. The same is here. The same cell line can have several TSSs, but whether the same cell can have several TSSs for one gene, we don't know. Well, maybe I don't know. Maybe you know. There are some single cell experiments, or whether there are capturing experiments in single cell done. I'm not aware of. Good question. So we have 3 questions to go, and then we are done. But you can, of course, give more questions. If I'm too tired to answer today, then we keep them for the next session. Following up question. In order to investigate how much a genetic mutation could affect the TF binding site, it is possible that such a mutation could affect multiple TF binding at that region. Would it be advisable to focus only on TFs based on their binding scores at that region? Right. As far as I understand, your question is that if the same place is occupied by several sites, right? And it happened that this place have a mutation in case of a particular disease, for instance, or mutation associated, or maybe it's not a mutation, but just a SNP associated with a disease. But anyway, it may change score or not of all of those sites which were found on that place. And we have, for instance, just let me show you. I'm sorry, just something. So again, to that story about that particular SNP in COVID, that, by the way, that SNP in the genome. And what we found here, you can see it. Oh, no, wait, here. So you see this software, this is a SNP, this position, which, I make it bigger maybe. Sorry, it's Word document. So this SNP actually affects this, I think this G, and then it in fact introduces its insertion. It's instead of one nucleotide, there's 2 nucleotides inserted, and then it leads. We call it site gain and site loss, and it can lead to creation of sites for 2 different factors. Well, both of, in this case, they are both from the same family, RF3 and RF8, but it can kill to lead to the site loss of these many factors. Which one is actually acting, we don't know. As I said, it maybe depends on the condition where this SNP is acting or giving the effect. But from a point of prediction, it may affect many binding sites for many factors, and it can create site or remove site. And in this case, we base our analysis on the difference of the score, but not only. Actually, of course, you can. What we do, we calculate the score of that site with a mutation for reference allele and for alternative allele, and we get 2 scores. and then we make a delta and between these two scores. And if delta is higher than, again, another cutoff, then we report this site. But you see the sites can be longer or shorter. And the score, if you look at the formula of the score calculation, it may be biased. It is indeed biased to a smaller site. So if site is small, to get the high score is easier than to get a high score for a longer site. So the scores, how we calculate them, they are not normalized to the length of the site. That's why we have another criteria here, which is a p-value. We calculate the p-value change, how this nucleotide change influencing the p-value, and the p-value for is more normal, is normalized. You can compare them to the different factors to each other and tell which one gets bigger effect. So among these 5, you can actually among these 6, you can say which one has bigger effect and which one has low effect, depending on which p-value changed more. Okay, so the co-occurrence of proposed by TRANSFAC is based on different experiments. No. So how valid the chances of? I'm a little bit the co-occurrence of proposed by so maybe it's not complete question. Somehow it was cut. But anyway, so I understand you're talking about co-occurrence of sites, and they are coming from different experiments. Right? Yeah. Maybe I understand your question. Again, so we are talking about co-occurrence of predicted sites. Of course, we can talk about co-occurrence of known sites, and then your question is right. So if this site was known from one experiment and another from one cell line and another site from another cell line, the co-occurrence makes no sense. And that's a valid question. But since we are looking for co-occurrence of predicted sites, then we assume that those predicted sites, they co-occur at co-function in the same conditions. That's why they co-occur, and physically how it happens, maybe you've seen those pictures of complexes when 2 factors are binding to the co-occurring sites, and then they have possibility to interact by another part of the protein, and to build a so-called activation domain. And people now calling it like a phase separation, place which leads to activation of a gene. So for that, you have to have these factors be in the same conditions. Okay, that may be repeating. The co-occurrence of TFBS proposed by Dan is based on different experiments. So what is the chances of co-occurrence actually? Right, that's what I'm answering. So that based on predictions. Finally, Alexander, thanks a lot for session. Thank you. Thank you. It was great. Many thanks. We'll be waiting for another one soon. I have more questions. Thank you very much for your questions. And yeah, it works. We're getting interesting questions from you, and we'll make these sessions from time to time. Actually, we plan to have at least one session a month. So please collect your questions. You can send them. There was a form on our website, or just send it to me. I guess when you entered this session, you left your email address. So we know your by email address, so we can send you all this recording, by the way, and also my email address and other information. So you will be not lost. Thank you very much. And thank you for your attention. And see you next time. Goodbye.